on? Yes. So I'm gonna switch gears to talk about poplars. Some of the speakers already make the point, so I don't think I need to belabor the importance of woody feedstocks for bioenergy initiative, other than pointing out that whether you're looking at southern pines or poplars, trees actually have always served multiple industries. So lumber, pulp and paper, and for energy use in the form of electricity has been the traditional use. So when you think about biofuels, or some speakers probably will talk about lignin carbon fibers, those are like new kids on the block. So for any tree improvement programs to be successful, we really have to be thinking about multiple use. I like this cartoon in particular because it hit my message home. If you can somehow manage to get biofuels out of trees while trees performing other services, that's really what we want to do. Okay. So why poplars? This slide says it all. As a genus, poplar trees are among the most widely distributed species in the Northern America, and that actually can be said for Northern Hemisphere. So wide distribution means rich natural variations that we can tap into for improvement, and also many other attributes that many other people already talk about, regardless of species. You want fast growing, you want easy propagation, and then also non-food species. It's been really important. Poplars have many firsts. Many of you probably don't know, actually the very first artificial forest regeneration plantation in the US was established with black cottonwood about a century ago. So soon after, large-scale breeding projects began, and poplar was also the very first trees to have tissue culture system developed, and then the very first tree to have the successful genetic transformation system developed as well. That led to many of the important discoveries, including transgenic trees with altered lignin characteristics. So in 1996, DOE actually chose poplar as the model woody crops for the biomass program headed by Jerry Tuscan at Oak Ridge. And that eventually led to the pitch to sequence poplar genome. An initiative started in 2002, and sequencing began in 2004. And the first tree genome was published in 2006. So we do have the blueprint to tap into the genomic advances as well. So primary use for pulp and paper, and then increasingly for bioenergy. So for that, the traits that we want for bioenergy actually are no different than what people have been wanting in the pulp and paper industry. We're talking about growth. Good growth is always a desirable trait. Shibu and many other speakers talk about stress tolerance, disease resistance that affect growth. When it comes to biomass utilization, cell wall traits are very important. And stress tolerance, marginal lands, and global climate change, the same message that you heard multiple times. And again, whether it's pulp and paper, biofuels, or other use of the woody biomass. So I'm gonna focus on cell wall traits and stress tolerance to give you a couple of examples to highlight the contributions of molecular biochemistry and genomic sciences to the biomass improvement. So cell wall traits have been the center of significant basic as well as applied research for a long time. And I guess I don't need to really tell you, as you can tell, the bulk of wood is composed of basically thick secondary cell wall portrayed here. Chemically speaking, you have cellulose, glucose, sugar chains that we like to use for extracting paper pulp and paper, as well as for biofuel conversions. You also have hemicelluloses, and then the phenolic polymers, lignin. They form, so the desirable component, cellulose, is embedded in those complex linkages, making them really difficult to access. So in the cartoon here, They don't really appear as those discrete components. So for pulping, in order to get access at pulp, you have to use harsh conditions, meaning a lot of alkalines and high temperatures. And in doing so, you lose the otherwise also useful hemicellulose sugar polymers components as well. So on the bioconvergence side, many speakers will elaborate on this later. But the principle is very similar. You have to apply some chemical treatment, although you can also use some other thermochemical mechanical pretreatment as well, and then tap into enzyme conversion to produce the fermentable sugars. And again, a lot of time lignin is the troublemaker, but I will argue that lignin actually it's probably the thing that can flip the coins around, because traditionally pulp and paper industry use the discharge of the lignin as energy source, but lignin can be developed into high value products like the carbon fibers that I showed you earlier, and also fine chemicals from there. But again, in the conventional thinking, lignin has been the troublemaker, and so since the pulp and paper industry days, there's been a lot of interest in understanding lignin. And this has set a really solid foundation on the lignin biochemistry pathway. So this was the pathway that I learned when I started the graduate school in the early 90s. But before I graduated, the pathway has evolved into something that we now call gridweight. We learn about many more enzymes, many more genes, many more of the subunits. And the discoveries continue even up to these days. New units continue to be discovered as recently as last year, new genes, new enzymes in the complex pathway. And so this is what we thought the primary pathway leading to the major lignin components in species like poplar. If you target the right genes, in this case, a gene called 4CL, at least in the laboratory, and this has been proven in multiple species and multiple systems, that you can knock lignin content down. As you've seen, this is actually a cross-section, the corner view of the cross-section. Lignin being the phenolics under UV light, it glows. So you see the blue colors here showing the wood part of the stems. You can actually knock the lignin content down, in this case, by 40%. The stem's still there. You don't see much of the lignin there. So this has been proven in multiple species, including in grasses and some other herbaceous species. We know what genes to target. It's what I call hotspots. If you hit any of those genes in this area, you're going to get lignin reduction. And this has been proven, as I say, a negative relationship between lignin content and pulp yield. You notice that I don't have any scale here. I just made up the graph because you can simply change the scale. It affects forage crop digestibility in alfalfa, for example. And then today, sugar yield as well. So it is a proven link between lignin content and biomass utilization. Although many people also work on structure alteration of lignin, the effect seems to be more species specific and not necessarily reproducible from labs to labs. This knowledge has now been taken to the field, so to speak, in a number of those large-scale association genetics projects. One currently underway at one of the DOE Bioenergy Center, Basque headquartered in the Southeast. So we have the knowledge. We can go out to the wild collection and screen those individuals for the extreme phenotypes. So the example here that the ones that have low lignin contents, those are breeding targets for perhaps bioconversion and pulping application. The ones with higher lignin content, they are valuable as well. They could be targets for thermochemical conversion. We can screen for the same traits, in this case, directly evaluating sugar release yield. Again, you can identify extreme phenotypes from field collection, verify their phenotypic behavior when you put them all together under one environmental condition, so-called common garden experiment. Or we can actually zero in at the gene level. In this case, the same gene that I showed you previously, shown by genetic engineering to affect lignin content. you can look for natural variations among the population and identify certain kind of DNA sequence variation that could lead to mutation that affect function, in this case, lignin biosynthesis. So with all the emphasis in dealing with lignin, sometimes we forget about or overlook the fact that lignin itself is very much an adaptive trait. So my lab has been working on a group of naturally occurring hybrids that originate from the two parental lines, the broadleaf from riparian habitats, more nutrient-rich, versus the narrowleaf from highland habitats, typically a more harsh environment. And the progeny hybrids segregate by various traits, including growth and secondary chemistry. So in a nutshell, in our conceptual model with all the data together, we tend to find this negative relationship between growth and lignin. So slow-growing clones tend to accumulate more lignin in the wood. It's also correlated to more phenolics in the leaves for the defense, for other response. So a trade-off between growth and lignin and some other undesirable compounds, it's probably a fact of life. So when we focus on our breeding, whether it's by traditional breeding or molecular breeding by new molecular genomics approach, it's really important to keep in mind that some traits vary consequences. So when we think about populating marginal lands, low lignin, fast growth, it's not necessarily the best suited phenotypes for perhaps long-term management. So they kind of give me, so basically growth, carbon allocation, nitrogen utilization from the soil are all integrated traits with the biomass characteristics that we like. So this lead into biomass improvement programs. Typically, if you're looking at pulp and paper industry application for poplars, the rotation is easily anywhere 6 to 8 years. For biofuels, we're looking at maybe 3 to 4 years, but those are long enough time to have issues that will be beyond our control. So from establishment to maintenance, diseases, drought, soil, nutrients issues, some other things that we talk about and you hear from other speakers vary from region to region. So the search for traits that could affect multiple desirable, the search for molecular targets, excuse me, that will affect multiple traits, it's been increasingly important. So the next case study I'd like to share with you, it's a trait affecting microtubules that my lab have been looking at for a long time. So instead of looking at those biological polymers, like I showed you about lignin and some other research on carbohydrate biosynthesis, we have been interested in the scaffolding of cell wall. And so shown in this tubular structure here, microtubules, they are thought to guide the position of cellulose, the components that we are interested in. And so we have always been interested in the role of microtubule in wood formation. I should remind you that microtubules, cytoskeleton components are everywhere. We have that as well in human cells. It's involved in cell division. It controls cell shape, cell expansion. And so this property of microtubules guiding cellulose deposition is specialized function in plant. There's a second specialized function of microtubules in plant. It affects gut cell behavior. So those kind of tiny white lines that you see here, gut cell is these tiny pores that you see on the leaf surface. So the opening and closure of the gut cells, it's really important for photosynthesis. Allow CO2 to come in, allow water vapors to go out. So that's also important component of microtubules. So my lab has been working on the microtubule components proteins for well over 10 years. It took us many tries to eventually figure out a way to produce transgenic plants that have perturbed microtubules. So the first thing that we look at, of course, is cellulose, right? Does it affect cellulose deposition as shown in this kind of textbook cartoon? Well, it didn't really change the cellulose. It didn't really change the hemicellulose, xylose, major components. It did, however, slightly affect lignin content and quite significantly, although still very small biologically, 10–20% of the lignin composition measured by the subunit fraction. We did find, however, that we see a trend of increase in some other so-called minor sugar components. And so we hook up with Michael Hans, whose lab at UGA, the Complex Carbohydrate Research Center, developed these beautiful cell wall glycone profiling techniques. Basically, you take the cell wall residues, you subject them through increasingly harsh condition, you take the extractables, and then basically scan through hundreds of antibodies raised against specific carbohydrate epitopes. So the long story short, actually I should mention that Siva, the postdoc who is working on this project, has a talk tomorrow, so you can hear more about the wonderful techniques. So if I highlight our wild type plants, the two transgenic groups here, you can see increasingly stronger colors here, showing increased signal affecting the so-called pectin, primary cell wall component. So this is something that classical model didn't even portray. You have microtubules, you have cellulose. In between, there's these primary cell wall components, this lignin composition that is affecting how cell wall scaffoldings are laid down. So it's a contribution on the basic research side. I mentioned microtubules' role on stomata behavior. So we put our transgenic trees through a series of drought experiments. And so when plants sense drought, one of the first things that they do is to basically close the stomata pore, so then you can conserve water. And by doing so, the dark gray color here is the wild type under well water condition. You compare with the white bars under drought condition. The stomata pore is closing, you have reduced the transpiration rates, that's what you expected. You have reduced the stomata gas exchange conductance, and you have reduced photosynthesis. Our transgenic plants, we perturb the stability of the microtubules. You see that photosynthesis, stomata conductance, and transpiration rates were pretty much the same under the so-called acute drought condition compared to well water condition. So under drought condition, the plants hold on to their stomata pores opening just a little bit longer. They can translate into more photosynthesis, a little bit more biomass yield. I mentioned the role of microtubules on governing cell expansion. We have yet one more surprising find from one group of the transgenic plants. If you look at the typical leaf shape of poplars, you plot the length with the width. One group of transgenic having wider leaves. And you can see along the developmental track, at some point in the development, we start getting much wider leaves. So this, again, for tree physiology, translates into leaf area, photosynthetic active leaf area. We look at the stems, and those are really young trees in a greenhouse, if you call them trees at all. But we did also observe increased stem biomass, which is attributable to increased stem diameter. So this is not a really common find. It was a really exciting find that you can find molecular targets potentially impacting a number of important biomass traits or properties. Biomass growth affecting both leaves and potentially stems, and also drought tolerance. And of course, I don't want to make it sound like this is proven and easy and all that, but this is sort of the core for a new project from the DOE Plant Feedstock Genomics program for us to dig in deeper for the mechanism and hopefully identifying additional molecular targets that can be used for breeding and for genomic selection for multiple trait improvement. So, in summary, I show you traits like lignin, cell wall traits, their causative effect between biomass utilization and cell wall components. In some cases, it's already well established. The know-hows, we know how to manipulate lignin, we know how to affect lignin composition, and the knowledge has been basically translated into breeding programs in many species. But these manipulation of lignin, it's really a balancing act. reducing too much lignin, you're gonna affect the structural integrity of the plant. The example that I showed you earlier, 40% reduction, it's possible in the labs, it's possible in the greenhouse. You put the plants in the field, they won't do well. Okay, so we know what is the limits for natural selections. And I mentioned this a little bit. I'm sure some speakers will follow up this chart. The lignin has been an energy source, but I will argue that they could be turned into higher value products to offset the more expensive type of biomass we're talking about here, being the woody biomass. When it comes to stress adaptation, it's increasingly more important, but it's an under-investigated area. Unlike the complex lignin, we don't really know 100% how lignin is made yet, but we have a pretty good idea. We have some candidate genes, but for stress tolerance, it's very difficult to pinpoint to any single gene and any single pathway. So they deserve a lot more investigation there, and that itself is a moving target given the wide coverage for initiatives like bioenergy and the different type of feedstocks that we need to have, the different kind of varieties within one given feedstock species. And I show you examples of biotechnological and genomics advances can assist in this effort, but despite the success with genetically engineered plants, the field trials to verify their behavior under more normal natural environment has been difficult because the procedure is getting lengthier and lengthier, and we are talking about just field trial to verify the finding, not to mention commercial deployment and all that. So there seems to be a really lengthy burden, so we can take the knowledge, turn to marker edit or genomic selection to identify similar traits that can be incorporated into breeding programs. And so the grand challenges, it's probably similar to many of that you already heard about. I will point out that tree biology research and development effort really is underinvested in this nation. And I talk to so many undergraduate students, I have so many bright undergraduate students who want to go into medical school. And you know, our education system, I'm talking about high school and college, plant biology really is underappreciated and undereducated. So not to talk about trees, right? So many students will even have their eyes pop open. You got genomes, you got all the resources. Everything you can do with human medical research, we can do with trees. You heard many times that we need flexible portfolio that is regional appropriate. We should consider multiple bioindustry markets. For the farmers, it's very difficult for them to put on an investment and not knowing whether or not the market will still be there 3 years, 7 years, 10 years down the road, and whether or not you might have other competing markets. Lumber price is always gonna be higher than pulp and paper, roundwood, than perhaps biofuels. And how do we deal with those kind of issues? It requires policies and many other factors. And I touched upon regulatory burdens for genetically engineered plants in general, for testing some of those traits and effects. And then sustainability, that Shibu did a really good job environmental services. We grow trees, of course we want to use biomass, but while trees are being grown, there are a whole host of other things going on. And those kind of service, we heard Ambassador Shine's talk, how do you include those into the market value of biomass species like woody trees that sometimes get unaccounted for, and you can start thinking about water filtration, windbreak, carbon sequestration, a whole host of other environmental and sustainability benefits. So with that, I will close by acknowledging former and current lab members, a number of collaborators for some of the projects that I show you, and then collaborators who share their data that I show you here. So thank you.
So questions for John Wright? I'll start out with one. you showed showed some evidence that there might be a linkage between your microtubule mutants and pectin or changes in pectin, which is not an expected sort of thing. Is that do you think there's a direct linkage, or is that perhaps indirectly linked to something else that might be going on?
The more we dig into, the more that people starting to find in the linkage. So in sickle mucilage, there is also a link between pectins and microtubules. So I think we are on the, we are just tipping the ice tips of, potentially something greater that will follow.
Other questions?
Yes.
Mesmerized. Thank you very much.